sometimes the traits have effect on other traits. So, what if we have tall plants and green plants and maybe dwarf plants and yellow plants? Would that play a role in the outcome? So, if we're looking at two factors in our Punnett square cross, this is going to be called a dihybrid cross. So, here we're looking at round yellow peas and wrinkled and green peas. And so we would look at two traits together. And so instead of just having one set of alleles that you would be looking at, maybe the big R, little r, now we're going to actually look at two traits together. So when he crossed two heterozygous plants, he resulted in lots of different variants. So round and yellow, round and green, wrinkled and yellow, and wrinkled and green. But you can see the recessive of both traits had very few compared to the others that were both. So when we do these Punnett squares, we're actually going to have multiple boxes. So let's take a look. All right, so we've got true breeding round and yellow and true breeding wrinkled and green. So we're going to say round is a big R. So this is going to be RR. And wrinkled, we'll use little r, little r. And then yellow and green, we'll use, Y's get a little crazy. Let's use, uh-oh. So this is going to be big R, little r, and we'll do G because that's a little easier to see. Okay, so what's the genotype of each parent? So the genotype, goodness, let's see here. I've already messed this up. So yellow, two big G's, and green, two little G's. Okay, so our genotype of each parent is going to be, for the first one, big R, big G, and then little r, little g. Now, what you're going to want to do is you're going to put, for the first one, we'll look at this one, you want every combination of the R's and G's on your top row. So we only have big R and big G. So in each spot, we're going to get a big R and a big G. So that's easy to figure out. And the same for the little r and little g. But what if, let's say we had, let me clear these off so you can kind of get an idea. Let's say the parent was actually big R, big G, little g. Okay, so in the first two boxes, you're going to put your first allele from the first trait. Then in the second two boxes, you're going to put your second allele from the first trait. Okay, so this guy goes here and here, and this one's going to go here and here. Now, your second trait, the first one, you're going to put in the first and the third, and then the second one, you're going to put in the second and the fourth. So that gives us all the possible combinations. So let's kind of look at that a little bit. Maybe get a different color. So this one goes here and here. This one goes here and here. Then this one goes first and third, and then second and fourth. Kind of like factoring in math, okay? So you want all the possible combinations. Now, back to the original problem. So we had, okay, RR and GG, and we had little. Okay, so since those are the same, two bigs, we're going to put R, G, in all of those. And our littles are all little, so little r, little g in each set. Now we're going to, just like we did before, bring both of these down and these across all the way. And remember, you want your capital letters to go first, but keep the same traits together. So we've got a big R and then a little r and a big G and a little g. And that's going to be true of every box because we're bringing a big R and a big G down in every box and a little and a little. So every single one of these, I can fill them all in, but they're all going to be big R, big G, little g, which means they're going to be totally heterozygous for round and yellow. 100%. Okay, so if the offspring from the above cross are allowed to self-pollinate, what are we going to get? Okay, so that's going to give us, on the last one, remember we got big R, and big G, little g on every single one. All right, this is where things get a little complicated. So remember we said to put our alleles on the box for the first parent, we put the first one on the first two, the second one on the second two, third one goes on 1 and 3, and the fourth one goes on 2 and 4. And then the second, he's going to be the same. So we can just kind of copy what we've got on the rows across. So R, big G, big R, little g, little r, big G, and then little. Now is where we're going to get a little bit more complicated. Okay, so the first box, I'm bringing down a big R and across a big R, and I'm bringing down a big G and across a big G. So that's crossing this one and this one. So going across here, bringing this one down, and again this one across. So big R, big G, big G. Okay, across and down, so big R, big G, big G. Okay, like magic, I've got this all filled in. Thought I'd save some time on your watching. So I filled this in, and you can see we've got lots of different combinations. So here's where it gets a little tricky. We have to write all of the possible genotypes and how many of each one. So we're going to just slowly work our way across, and I'm going to use some different colors to help keep this straight. So for the first one, we're just going to go across the boxes. We've got big R, and then, you know what? I know I'm going to need to make this a little smaller. So let me try that again. Okay, big R, big G. And how many of those do we have? So we've got, I'm going to cross them out. We've got one of those, and I don't see any more. So we're just going to put 1. Now we're going to look for this. We've got big R, big G, little g. So we've got one of those there, and there's a second one. And I don't see any more of those. So we've got 2 of those. Now let's do This box, we've got big R, little r, big G, big G. So do we see any more? There's one, there's two. I think that's all. So we've got two of those. Now if we look at this one, we've got big R, big G, little g. So we've got 1, 2, 3, 4 of those. Okay, working on our way on. We've got, this one's done, so now we're here. Big R, little g, little g. So there's 1 of those, and I think that's all. So now we'll look at this one, big R, little g, little g. So there's 1 of those, 2 of those. And then we've got, here's our next one, little, big. And I only see 1. Then we've got little, big little. So there's 1, 2 of those. And then our last one, little, 1. And that gets all of them. So just to double check ourselves, if we add up all of these numbers, we should have 16. So those are our genotypes. Now our phenotypes, the possibilities, we've got round, and yellow. So the first one here, this is round and yellow. Because remember, big G was yellow. This is round and yellow. So any big R's and big G's we want to check off. Okay, so it looks like we've got 4, 6, 8, 9 round and yellow. Then if we look at this one, he is round and green. So round and green, this is round and green. So it looks like we've got 3 of those. Then our next one here, we've got wrinkled and yellow. So that would be this guy and this guy. So 3 of those. And then the last one, this one is wrinkled and green. And we've got 1 of those. So again, these numbers should add up to 16 because it's how many boxes we've got. All right, so that's probably the most complicated is when you have heterozygous and heterozygous. All right, so those are some dihybrid crosses.